Angiotensin blockade in late autosomal dominant polycystic kidney disease. Background: hypertension developed early in patients with autosomal dominant polycystic kidney disease and is associated with disease progression. The renin-angiotensin-aldosterone system is implicated in the pathogenesis of hypertension in patients with ADPKD. Dual blockade of the RAAS may circumvent compensatory mechanisms that limit the efficiency of monotherapy with an angiotensin-converting enzyme inhibitor or angiotensin II receptor blocker. Method: in this double-blind placebo-controlled trial with redoubling assigned in patients 8 to 64 years of age with ADPKD. With these dose adjusts to achieve a blood pressure of 110 to 7 to 138 mmHg. The composite primary outcome was the time to death, end-stage renal disease, or a 50% reduction from the baseline estimate GFR. Secondary outcomes include the rate of change in urinary aldosterone and albumin excretion, frequency of hospitalization for any cause and for cardiovascular causes, incidence of plaque, frequency of ADPKD-related symptoms, quality of life, and adverse study medication effects. Patients were followed for 5 to 8 years. In results, there was no significant difference between the study group in the incidence of the composite primary outcome. The two treatments controlled blood pressure and lowered urinary aldosterone excretion similarly. The rate of decline in the estimated GFR, urinary albumin excretion, and other secondary outcomes and adverse events, including hyperkalemia and acute kidney injury, were also similar in the two groups. I imagine about the topic and conclusion. Monotherapy with an ACE inhibitor was associated with blood pressure control in most patients with ADPKD and stage 3 chronic kidney disease. The addition of an ARB did not alter the decline in the estimated GFR. The video.
Renin-angiotensin-aldosterone system. The renin-angiotensin-aldosterone system is a classic endocrine system that helps to regulate long-term blood pressure and extracellular volume in the body. The system begins with the release of angiotensinogen into circulation by the liver. This may be in response to low blood pressure and adverse changes in sodium concentrations. An enzyme, renin, is secreted which cleaves angiotensinogen to form the inactive decapeptide angiotensin I. Further transformation of angiotensin is carried out by angiotensin-converting enzyme, or ACE. This is predominantly found in the pulmonary circulation. However, ACE is also produced in the vascular endothelium of many tissues, including the kidney, adrenal gland, brain, and heart. The angiotensin-converting enzyme converts the inactive precursor angiotensin I into the vasoactive peptide angiotensin II. In addition, alternative pathways exist that do not rely on either renin or ACE. In non-renin pathways, enzymes like tonin and cathepsin D release angiotensin I from angiotensinogen and tissue plasminogen activator, or TPA, and can make angiotensin II directly from angiotensinogen. This bypasses the midway production of angiotensin 1. Enzymes like kinase can form angiotensin 2 from angiotensin 1 via an ACE-independent pathway. Angiotensin converting enzyme also degrades bradykinin, which is required for synthesis of a major vasodilator, nitric oxide. Angiotensin 2 binds AT1 receptors expressed on the surface of vascular endothelium and impairs nitric oxide synthesis as well. Reduced bioavailability of nitric oxide combined with the stimulation of AT1 receptors on smooth muscle cells causes vasoconstriction. In addition to a vasoconstriction effect,